Hello everyone. In our last session we have discovered that the body has the ability to distinguish self from non-self. This is due to the presence of biological markers on the membrane of our body cells. The first biological marker so far discussed was the HLA marker, which is found on the membrane of nucleated body cells. Today we're going to discuss another self marker. As you would observe here, the blood groups show another self marker. These markers are found on enucleated body cells, which are the red blood cells. In this session, the following points are going to be discussed. First, we're going to identify the markers of self in the enucleated cells, that is, in the red blood cells, as the agglutinogens, which are the blood group antigens. We're going to recall the different agglutinogens and their corresponding antibodies, or agglutinin. Agglutinin is the blood group antibodies, and agglutinogens are the blood group antigens. The notion concerning blood group antigens and blood group antibodies have been discussed in grade 8. We are also going to study the consequence of a blood transfusion between incompatible blood groups. The meaning of incompatible blood groups, that is, blood groups that are not going to get along during a blood transfusion and which would obviously lead to complications. We are also going to learn how is the possible blood group of an individual determined. Finally, we're going to identify the presence of another self marker on the red blood cell apart from the ABO system, which is the rhesus system, that is, the rhesus factor constituting the rhesus system. The differences between the ABO system and the rhesus system is also going to be clarified. Let us proceed with our first point concerning the blood group agglutinogens and the blood group antibodies, or agglutinin. As I have clarified earlier, the nucleated cells have the HLA markers on their surface. On the other hand, the enucleated cells, that is, the red blood cell, they're going to have their own markers, which is going to constitute the ABO system. The answer to this question here, what are the markers of self in these enucleated cells, that is, in the red blood cell, is now clear, which is the agglutinogens or the blood antigens. Now, where do we find the blood antigens or agglutinogens? On the surface of the red blood cell, on the membrane, we're going to find certain molecules which are called glycoprotein molecules. They are carbohydrates attached to protein molecules. This is why we call them glycoprotein. These glycoproteins are going to constitute the antigenic or the agglutinogenic determinant of the ABO system. They're going to differ from one individual to another, and they're going to determine our blood group. What are the two different antigens or agglutinogens? They are antigen A and antigen B. What are the blood antibodies or agglutinin? They are anti-A and anti-B. The antibodies are found in the plasma and they are innate. Innate means that they are inborn, found at birth. Let us discuss the different agglutinogens and their corresponding antibodies and the blood groups. As you know, we have 4 different blood groups, which are Group A, that is type A, B, AB, and O. In Group A, the red blood cells are going to show the marker here, which is antigen A. Note the structure of this marker. In the plasma, we're going to find the antibodies. The antibody here are the anti-B antibodies. Notice that the structure of the antibodies here, the anti-B antibody, are not complementary to that of the antigen. That is, this part here, which is referred to as the antigen binding site, and this notion is going to be discussed later on in the coming sessions, this part, this antigen binding site, is not complementary to the antigen. They are different. In type B, or Group B, the red blood cells are going to show a different agglutinogen, a different marker, which is the antigen B. Note the shape of the antigen here is different from that found in Group A. In the plasma of type B, we are going to find the anti-A antibody. Here too, the structure of the anti-A antibody in the plasma of Group B is not complementary to the antigen. On the other hand, individuals with type AB show antigens A, both antigens A and antigen B, on the membrane of the red blood cell. However, in their plasma, we're not going to find any antibody. That is, they have neither anti-A nor anti-B antibodies. Individuals with Group O have neither antigen A nor antigen B on the red blood cell. They have no antigen. However, in the plasma, they're going to show that they're going to have both anti-A and anti-B antibodies. So, as you have observed, Group A has antigen A, but has anti-B antibody. Group B has antigen B, but has anti-A antibody. Group AB has both antigens A and B, but has no antibodies. Group O has no antigens, but has both anti-A and anti-B. Note that the blood groups are named according to the antigens they are representing. Antigen A, Group A. Antigen B, Group B. Antigens A and B, Group AB. No antigen, Group O. So, in conclusion, we will never find in an individual's blood the antigen and the corresponding antibody. The question that arises here is, why can't we find in an individual's blood the antigen and the corresponding antibody? In order to find an answer to our question, let us have a look at the figure which is shown here. The red blood cells have antigen A on their membrane. These are antibodies which are the anti-A antibodies. The antigen binding site is complementary to the antigen A on the red blood cell. When these are placed together, it is going to lead to clumping of the red blood cells. These are the red blood cells, they are clumped together, that is, they are grouped together. This is agglutination, also referred to as hemagglutination. What has actually happened is that the anti-A antibody binds to the antigen A on the red blood cell and pulls them together in a group like this, which is referred to as a clump of red blood cells. This agglutination, also referred to as hemagglutination, is going to lead to lysis or destruction of the red blood cells, which is also called hemolysis. Next, we're going to study the consequence of a blood transfusion between incompatible blood groups. In order to find the cause of a blood transfusion failure, the following experiment was carried out, where the red blood cells of an individual was mixed with the serum of another. The results are shown here. The negative signs indicate an absence of hemagglutination, and the positive ones indicate the presence of hemagglutination. In the first and in the fourth cases, there was no agglutination. When the red blood cell of group A was mixed with the serum coming from the same blood group A. Likewise, when the red blood cells of group B were mixed with a serum from group B, no agglutination was also observed. In contrast to the second and the third case, where agglutination was observed when the red blood cells of a group was mixed with the serum of a different group. That is, group A, red blood cells from group A were mixed with the serum of group B. And here too, the red blood cells of group B were mixed with serum from group A. So what information can you draw out from document B? The information that you would draw out here is that agglutination will only happen when we mix the red blood cell of a given group with the serum of another group.